So here we'll discuss the differences between genomes and genes and review how the genome is organized and cover the basics of DNA structure. So the genome is the entire genetic complement of an organism. It's stored in DNA in most cells, but sometimes as RNA in some viruses. And the entire genome is present in just about every cell. And every cell that does have a genome has roughly the same genetic complement within a species. Although there are some exceptions to this, and they're sometimes referred to as genomic instability. The entire genome is fully duplicated just before cells divide, so that way each daughter cell can also have a full genome. And the genome is stored in DNA in chromosomes. And a chromosome is defined as a single molecule of DNA and all of its associated proteins. So now if we contrast that with a gene, individual gene is a much smaller segment of DNA, and it's just a specific DNA sequence that contains the instructions or the code for the production of a specific protein. So many genes, plus lots of other non-coding DNA sequence, interact with proteins to make up an individual chromosome. And many chromosomes make up the entire genome. And humans, for example, have 23 pairs of chromosomes in their genomes. So we said that individual genes code for specific proteins, and this idea that genes in the form of DNA are transcribed into RNA, which is then translated into protein, is known as the central dogma of molecular biology. So a gene is a specific sequence of DNA that's made up of these colorful nucleotides, and the human genome contains somewhere around 25,000 individual genes. The DNA in that gene is transcribed into an RNA molecule, which acts as an intermediary and carries the information from the DNA to the ribosome. And at the ribosome, that RNA is translated into a sequence of amino acids that make up a specific protein. So let's first start by contrasting the ways that genomes are organized in prokaryotes like bacteria versus how they're organized in eukaryotes. So in prokaryotes, the arrangement of the genome is pretty simple. There's generally only one chromosome per cell, and that single chromosome contains the entire genome of that species. So the prokaryotic genome is generally small, around 10 to the 5th or 10 to the 6th base pairs. Often the prokaryotic chromosome is circular, but not always. And there's very little of that extra or non-coding DNA. Now let's contrast that with the typical eukaryotic genome. So eukaryotes, even like single-cell eukaryotes, have much larger genomes than prokaryotes. The human genome contains about 10 to the 9th base pairs. In eukaryotes, instead of having a single circular chromosome, the cells contain DNA that's organized into multiple linear chromosomes. And eukaryotic organisms that use sexual reproduction have a duplicated genome after reproduction. So germ cells like sperm and oocytes contain a single copy of the genome, and this is known as haploid, while somatic cells, which is just all the other cells other than the reproductive cells, contain 2 copies of the genome, which is known as diploid. So in eukaryotes, unlike prokaryotes, there's lots of extra DNA. So there are large expanses of DNA in between the genes, which are known as intervening sequences, and interrupting the genes, which are known as introns. So in humans, a very small percentage of the genome actually encodes proteins, somewhere around maybe 1.2%, while the rest of it is this extra or non-coding DNA. Some of the extra DNA does play a role in regulating gene expression and protein levels, but the function of much of it is completely unknown. And finally, eukaryotes also have small, circular, prokaryotic-like chromosomes with different sets of genes in organelles like mitochondria and chloroplasts. And we said the entire genome had to be duplicated at every cell division throughout development of the organism. So every cell in the mature body essentially has the same total genetic material. So there's lots of DNA replication happening in our cells, and it's really important that the accuracy of replication or fidelity be extremely high. So let's briefly review the structure of a DNA molecule so that later we can better understand how DNA is replicated. So DNA has this double helix shape, which is like a ladder twisted into a spiral. The sides of this ladder in blue are a sugar phosphate backbone, and each step or rung of the ladder is a pair of nucleotides. So in this image, we can see the locations of the major groove and the minor groove in the DNA molecule. So the minor groove falls between these two sides of the ladder, and the major groove falls between the two turns in the helix. And so this is an important difference because certain proteins that bind to DNA to alter its structure or to regulate transcription or replication bind only along this major groove. Now, if we unwind the helix and zoom in on just a few base pairs, this is what we would see. So here we're looking at many nucleotides linked together with that sugar phosphate backbone in blue and the paired bases in between. So an individual nucleotide molecule is made up of deoxyribose, which is a type of sugar with 5 carbon atoms, a phosphate group made up of phosphorus and oxygen, and a nitrogenous base. And so DNA is made up of 4 types of nucleotides. There's thymine, adenine, cytosine, and guanine. Thymine and cytosine are pyrimidines, and they have only this one ring, while adenine and guanine are purines, and they have 2 rings. And so phosphodiester bonds connect this backbone, and the rungs of the ladder that are made up of these 2 paired bases that connect in the middle, and those bases are held together by hydrogen bonds. So these dotted lines are hydrogen bonds. And thymine and adenine can pair up because they make 2 hydrogen bonds, and cytosine and guanine can pair up, and they make 3 hydrogen bonds, and these are what are known as the Watson-Crick base pairs.